안녕하세요. My name is CHAWAPOL BOONKOET. So let's start with the simple questions. So if every year 30% of our jasmine rice of Thailand is lost because of a rice blast disease, how would it affect the farmers, our economy, or even our rice on our dinner plate? Bringing my idea on applying CRISPR-Cas9 to develop blast resistance in Thai jasmine rice, KTML 105 by focusing in Thailand. The importance of rice in Thailand. Thailand is one of the most world's top rice exporters. So in 2023, Thailand is the second largest exporters of rice in the world, according to OEC. And KDML 105, for those who don't know, KDML 105 is cow dog mali 105. It's a type of Thai jasmine rice. KDML 105 is famous for aroma and quality. This is the awards that given to Thai homily, the jasmine rice. And bringing the problem of rice blast disease, it is caused by a fungus named Magna orte oryzae. It's giving the yield loss of 10 to percent of rice production cause farmers to lose income. And in 2019, there's a reporter, the THAILAND, it's a news saying that it is severely affecting the rice in Thailand from the rice blast disease for 480 million square meters. So the current solutions, you can use fungicides, but it is costly and it is negative to environmental impact. Biomagnification, and it can develop themselves, which is pathogen resistance. And for traditional breeding, it is time consuming, and you can get undesired traits from the methods. For the literature review, from Mr. Wang and his researchers team, demonstrate that you can use CRISPR gene editing on the rice, which is they edited the OS-ERF922, which success. And for Mr. So and his researchers team, they developed a modifying the genes of ERF922, PI21, and BSRD1, which is multi-editing genes. Yes. For the methods, I'm gonna use the Thai jasmine rice from the northern east Thailand because it is the most affected areas from the disease. And for the DNA preparation, I will extract the DNA from the KDML 105 leaves. And for the CRISPR-Cas9 editing, I will design the guide RNAs targeting these genes and use Cas9 enzyme to cut DNA at the target sites and introduce edits through transformation methods, which is agrobacterium mediated transformation. And for the validation, I'll check edited sequence by PCR and DNA sequencing, grow edited plants and breed to the next generation. And for the resistance testing, I'll expose edit plants on magna porte orice under controlled conditions and evaluate resistance by comparing disease severely against wild-type KDML105. And for field trial, if it is successful, we will test in the real field. And why CRISPR-Cas9? So it is precise, faster, and it can keep aroma and quality. And CRISPR-Cas9, it is not transgenic because it's worked by modifying the plant's existing DNA. For expected results, it shows fewer blast symptoms. It maintains grains, aroma, quality. For benefits, farmer got higher yield, environmental got less chemical pollution, and for the economy, we can maintain our jasmine rice exports values. For the conclusion, CRISPR-Cas9 is a strong candidate from solving the blast disease, and this new method hasn't been yet tested on KDML105, and it can strengthen our Thailand's agriculture and farmer livelihoods. Thank you. This is the reference.
Thank you for the very interesting topic. Koreans probably never heard about. So you said knocking out the one specific gene from this jasmine rice, right? So is it the gene function is really characterized? You know what is the gene function in the rice? What does the gene really do?
The gene functions?
Yeah, gene function. What's the role in the rice?
Can you explain more?
So when you knock this gene out, you are going to expect the rice is going to be resistant to this fungal infection, right?
Yes.
So what is the function of this gene? So is this gene is going to make the rice sensitive to this fungal infection? Then what is the mechanism?
So from using the
So you are trying to knock out this gene because this gene gives the rice the sensitivity
Yes.
to the infection, right?
Because
So what is the mechanism? How this gene confer the sensitivity to the rice?
So there's for example,มันเอากลับยังไงครับ For example, in Mr. Wang, he tested on another kinds of rice species. He found that OSERF922 it is a gene that makes rice sensitive. So he used the CRISPR gene method to change the OSERF922 out and make it replace the new one, cut out and
okay.
Yeah.
You are going to replace the gene.
Yes.
Okay. Then this gene probably really maybe dispensable for the life of the rice, maybe.
So Mr. Wang, he tested the normal of how is it affect on can they live on the real field? He tested and it is no problem with it, and he tested many generation T1, T2, T3, and there's no problem with it.
Okay, I have another question about what you just mentioned. So this gene knockout worked in other species of rice. Is that what the paper talks about, do you think?
Sorry.
So when the authors knock out this gene and then replace with a new gene, so that can confer the resistance to the fungal infection, right? Is that what the paper talks about?
The papers talks about using CRISPR gene editing on the rice.
Right. Is that paper also talks about the resistance to the fungal infection,
It
or
is-
just gene editing?
It is talking about how genes editing affect the grain's qualities and
Okay,
Yeah.
not about the resistance. Okay.
yeah.
Okay, thank you very much for the interesting talk. So after gene editing with this agrobacterium transformation, so what do you expect in terms of this mutation? Is it going to be one allele or two alleles? Basically, is it going to be heterozygous mutant or homozygous mutant after gene editing?
Can you
So do you understand my question? So after CRISPR-Cas9 application, so what do you expect? Do you think the gene editing is going to happen in one allele, or is it going to happen in two alleles? So I mean, you are going to breed and to obtain the homozygosity. So I think it's going to be heterozygous, but I'm just asking whether you understand this technology.